Murray-Darling Freshwater Research Centre has had a project team working on condition monitoring in the Living Murray program for up to the last 8 years at different sites. We've got condition monitoring sites across the Hattah Lakes Icon site and also across Lindsay, Mulcra and Wallpolla Islands, which is part of the Chowla Icon site under the Living Murray program.
The Living Murray Condition Monitoring Project reports on icon site condition and informs management on the prioritisation of environmental water.
Our project team are involved in monitoring a variety of components at the icon sites that we visit.
We monitor tree condition, we monitor tree population viability, we monitor lignum condition, we monitor the distribution of Kumbungi within the waterways, fish communities, floodplain and wetland vegetation communities.
I look at wetland vegetation, floodplain vegetation and lignum understory. To sample wetlands, we set up usually 4 transects at each wetland, and then we do 1 metre by 1 metre quadrats 15 times at each of the transects to try and pick up different vegetation at each of the elevations on a wetland. It doesn't matter if the wetlands are dry or wet, we sample them whether they've got water in them or not, because there's different vegetation communities that come out of that. Higher up on the floodplain you'd probably find a lot more dry stuff like sclerulinas, salt bushes, atroplexes, that kind of thing. Lower down on the floodplain you might have more stuff that grows in terrestrial damp conditions after a flood. Lignum is seen as ecologically significant because it provides habitat for lots of fish during times of flood and birds when it's not flooded. We look at that separately to wetland understory or floodplain understory. We have a number of different sites, and at each of the sites we have 30 different plants that we have a look at, and we assess them visually for colour and for viability as well.
We monitor tree condition using a range of variables. Probably the most important one would be the canopy condition. Crown extent is essentially the foliated proportion of the crown, and crown density is basically just how bushy that foliage is. There's a crown condition target for river red gum and there's a crown condition target for black box. The program captures the response of trees to both environmental flows and natural flows.
We measure the Kumbungi along a number of reaches throughout Lindsay, Mulcra, Wallpolla and also down at Hattah to measure the distribution of Kumbungi throughout that area. We measure the Kumbungi distribution annually just to see whether it's expanded or shrunk in areas, and sort of to relate that to the hydrology of the area as well. Kumbungi is a native plant in the river system. In most cases, Kumbungi is beneficial in low numbers, and it can be habitat for native birds and fish. It likes stable water conditions, so when the water conditions are kept stable, which is sort of an unnatural state, then Kumbungi can expand and start to dominate areas, outcompeting other native plants that can also clog up waterways. With the understory vegetation, if we're unable to ID the plant in the field, we collect it and take notes and bring it back to the lab, where we try and ID it using keys and also eventually put it in our herbarium.
So the bird surveys are done by Sunraysia bird observers, and they take a snapshot of bird assemblage at each icon wetland. They play a really important part to the ecosystem, and they're also an indicator of the condition of the environment. So we break up the water birds into 5 functional groups. So we look at the water birds annually to record assemblages year to year. Depending on how much water is in the wetland, you'll get different species at different times of when the water's receding. So we're looking for breeding activity and also breeding habitat as well. So at the moment we get a lot of Australian pelicans, and they're piscivores. They feed on fish, so it's a great indicator that there's fish in the wetlands too when they're there. It's great to see great egret and pied cormorants because they're less common.
My main focus has been fish community monitoring and tree assessment. Fish sampling for the Living Murray is undertaken annually. We use a number of methods to capture the fish. We use electrofishing, and we use different netting techniques, including small fyke netting, seine netting and bait traps.
So Billabong's wetland is important for fish habitat. They can be fish nursery, so when a flood comes through, baby fish can come into it and grow up. When larger fish can use it for habitat and food source. So here today we caught a fish, so basically we measure it with standard length, the total length, and then we do a weight and then do a general condition on it, so we look for any diseases.
When we survey the fish, we're looking to see population structure, so to see if there's any juvenile fish being recruited into the system, and we're also looking at fish abundance. So the main species of fish that we encounter through this area are Murray cod and yellow belly and bony bream. Some non-native species or pest species that we capture are carp, goldfish and eastern gambusia. The biggest fish I've seen caught was $1.25 m, a Murray cod, and that was pretty exciting.
I'm primarily looking for a score for the condition of the icon site. So how each of those components contributes to the overall condition of the icon site at any point in time.
The Living Murray is a program that's funded through the Murray-Darling Basin Authority, and we work closely with the Mallee Catchment Management Authority and also the Victorian State Government in carrying out and communicating the results of the monitoring at these sites.